Hello, this video is based upon the paper Using Punnett Squares to Facilitate Students' Understanding of Isotopic Distributions in Mass Spectrometry. This article was published in the Journal of Chemical Education in 2006. Punnett squares are widely used in biology to predict the results of crosses. So, for example, if we have a genetic cross between two parents, we might have a situation where we have both parents are heterozygous in some particular trait. And the way that we represent that for each parent is the dominant allele is given a capital letter, so we have capital A. The recessive allele is given a small letter. So, and since they have both a dominant and a recessive allele, we say that they are heterozygous. So if we're looking at the cross between two heterozygous parents, we can write it as big A small a times big A small a. Okay? And then we represent it in a square as such. And we separate the alleles into individual columns or into individual rows, depending upon which parent we have. And the key idea here is that the probabilities of each of the parents sending out a sex cell with a particular allele is exactly 50%. It's a 50% chance of having the capital A and a 50% chance of having the small a. Same for example, this might be the mother and this would be the father. And then when they combine to form any of the progeny, the children, they get one allele from one parent and one allele from the other. We can write down at the intersection of each row and column which combination of alleles the child will receive. So for the first row, first column, we have big A and big A, so we write down both A's. So they're going to get two copies of the dominant allele. For the first row, second column, we have capital A, small a. The order of the alleles doesn't matter. For the first column, second row, again we have a capital A and a small a. And then at the intersection of the second row and second column, we have two small a's. And there are a couple different ways to interpret the results that we get here. Now we do feel that the probabilities are proportional to the number of boxes in this case. So we see that we have the likelihood of having AA is 1 out of the 4 boxes, so that probability is 1/4. The probability of having large A, small a, we see that we have 2 boxes out of 4, so the probability is going to be 2 out of 4. We also know that's 1/2, but we'll just write it down how we get that. And then last but not least, the probability of having 2 small a's is going to also be 1 out of 4. And since we must have some result, if we add up the probabilities of 1/4 plus 2/4 plus 1/4, we see that we get a total of 1, is what we have to get for any particular cross. In terms of the phenotype, the appearance of the offspring, we notice that since we have a dominant and a recessive gene here, we will exhibit the dominant trait if we have 2 dominant alleles. So AA will look like the dominant expression. Capital A, small a will also look like that because big A is dominant over small a. So 3/4 of the progeny will appear like the dominant trait, and only 1/4 will appear with the recessive trait, obviously. That's what we call the phenotype, the appearance of the offspring. In terms of the genotype, that's what we have down here. So we have 3 different genotypes. We have the homozygous dominant, the homozygous recessive, and then we have the heterozygous situation here. Now, it's well known that for many particular traits, even in the cases where we have the one allele of dominant and one allele of recessive, that even though this is going to exhibit the dominant trait, there is often what is called a heterozygous advantage, that there's often an advantage for organisms that are heterozygous for some particular trait. The situation of a cross between two organisms that are heterozygous for a particular trait actually bears resemblance to a situation that we encounter in chemistry, particularly in mass spectrometry. And this situation is if we have a molecule that has exactly two atoms with bromine. And we know that bromine exists in two different isotopes. There's bromine 79 and bromine 81. So this would be one of the bromine atoms, and the second bromine atom we'll put along the side. And it again can be bromine 79 or bromine 81. So what's interesting here is what combinations we're going to encounter in actual molecules. What is peculiar about the isotopic distribution for bromine is that the probability of getting a bromine 79 is almost exactly the same as for bromine 81. So in a low accuracy sort of sense, it's kind of a quick and dirty, we can assume that these probabilities are exactly equal. So that the situation in its own way resembles that of the cross of the heterozygous traits. So we can almost think of the bromine 79 as being the capital A, the bromine 81 as being the small a. And then we can write it out as a cross, so just kind of emphasize the Punnett square crossing aspect to it that we had just seen. And the situation, the probabilities work out almost exactly the same here. So if we just assume that the probability of 79 and 81 are exactly the same for the time being, then we get that the probability of having AA here. So A, what does AA really say? AA is having two bromine atoms that are both bromine 79. And we had seen previously from the previous Punnett square that this particular probability is 1 in 4. For the capital A, small a, the heterozygous case, what does that mean? Well, it means that we have one bromine 79 and one bromine 81. And the likelihood of this particular combination occurring is going to be 2 out of 4. I'll write it as 1/2 here. So that's the probability that is 50%. Let's kind of write this in terms of decimals. That's 0.25 if you write it as a decimal. This is 0.50 as a decimal. And then the last combination, the homozygous recessive case, is the equivalent of having two bromine 81 atoms in your molecule. Exactly 2. And then the probability of that is also going to be 1/4 equals 0.25. The way that we can recognize which situation we have in mass spectrometry is by the total mass, because the mass here of two bromines is twice 79. So we have 158. In the case of 179 and 181, we have a total of 160. And then if we have two bromine 81s, the mass is going to be 162. But even more than that, we would recognize that typically we don't look at the total mass. What we're really interested in is the mass of the molecular ion. So if we think of it in terms of molecular ions, we can write the same sort of box here for bromine. And I will write this again to the top to remind us. But we'll use the same labels for the rows, so I don't have to write it again. And we notice that this combination here of two bromine 79s would correspond to the molecular peak, which we refer to as M in mass spectrometry. If I have 179 and 181, that's going to give me a mass 2 units greater, so that's going to be M plus 2. For an 81 and a 79, since we can't tell which bromine is which, the order doesn't make a difference here. So that's also be a combination to be M plus 2. And then if I have two bromine 81s, that gives me a combination that has a mass of M plus 4. Ultimately, a Punnett square is just a multiplication table. So we can make use of this idea of the Punnett square as a multiplication table to get an even more accurate assessment of the probabilities of particular isotopic distributions. So to do that, we're just going to draw the same box that we did before. I'm just going to draw a little bit bigger here. And we're also going to label each column or row with the probabilities. So the actual probability of having bromine 79 in a sample of bromine is actually 0.5069. So it's slightly more than 50%. The probability of having bromine 81 is going to be 0.4931. So we see that in a crude sense how close this actually is to a 50-50 or a 1-to-1 combination. So it is very close to the idea of having a heterozygous trait. And then for the rows, again, so this is bromine 79 here, and this is bromine 81. Now that we've labeled our table in this way, we can more easily exploit the fact that we have what is essentially a multiplication table. And to figure out the probabilities of each combination, I merely multiply the value in the row times the value in the column. If I do this, I get kind of round it to 4 decimal places. So what I've done here is just to multiply the value for the column times the value for the row, and then we put that in a box. The only thing we have to keep track of is recall that this box here and this box here correspond to the heterozygous situation. So these both correspond to one bromine 79 and one bromine 81, so that when we're assessing the overall probabilities, we have to make sure that we add these two boxes together. So what we get, if we do it and do some red to kind of highlight it, again, so if we're looking at two bromine 79s, the actual probability is 0.257. So it's slightly greater than the 1/4 that we had done previously. The probability of a bromine 79 and a bromine 81 is going to be these two boxes combined, so that's going to give us 0.5000. And last but not least, the probability in the second row, second column, is for two bromine 81s in our molecule, and that's going to give me a probability of 0.2432. And recall that we can correlate the probabilities of a particular molecular combination of isotopes by heights in mass spectrometry. So the probabilities are proportional to the heights. So we see here that we see a ratio of 1 to 2 to 1 of the peak heights for a situation where we have the M plus 2, and M plus 4 peaks. In mass spectrometry, we always want to look for where the molecular peak is, and we also want to see if there are two significant peaks or more at the very high end of the mass scale that are separated particularly by two mass units. Because when we see the separation by two mass units, it reminds us that the two different isotopes of bromine are separated by two mass units, and in the case of chlorine, the two isotopes are separated by two mass units. So that is one of the first things we want to look for when we're analyzing mass spectrometry. The first thing we always look for is the 91 peak, because that tells us about trillium. But then the second thing we would want to look for is this separation by 2 units, and that is often indicative of the presence of a halogen such as bromine or chlorine in our molecule.
Here we have a 2 by 2 Punnett square showing the situation of having exactly 2 bromine atoms in the same molecule. You can imagine this is the column for bromine 79. This is the column for bromine 81 for the first atom. For the second atom, the first row is bromine 79, and the second row is bromine 81. In this case, the white box corresponds to the probability of having 2 bromine 79s. The darker gray corresponds to the situation of having 2 bromine 81s. The other situation where we have 1 bromine 79 and 1 bromine 81 is shown in light gray. So if we rearrange the boxes, we can actually look at the relative probabilities, because we have to add the probabilities of all the boxes that have the same colors. So we see that for the 2 79s, 1 and 4, the probability of having 2 81s is approximately 1 in 4, and the probability of having 1 79 and 1 81 bromine atom is 1/2, or 50%.
We can extend our previous ideas of making the height and width of a particular column exactly proportional to the probabilities. So instead of these being exactly equal in size, this one is about 0.50 and this one's 0.49. Then if we do it 2 by 2 dimensions, we get that the areas for a 2 by 2 cross will be proportional to the probabilities. We can extend that idea to 3 dimensions, and now we make the height, width, or depth of a particular column proportional to the probability of either bromine 79 or bromine 81, and we can determine the probabilities for all the combinations of 3 bromine atoms in one molecule. A molecule for which this might apply would be bromoform, CHBr3, and we would like to assess the relative probabilities of all the combinations of the isotopes. So we can make a cube. So we have, for the columns here, this is column for bromine 79, bromine 81, bromine 79, bromine 81, and then going into the back, the near section is going to be bromine 79, and then bromine 81. And we notice that if we do this, we get a number of blocks that we can color in. So for example, this block here, this one first, we want to leave open, but this is for 3 bromine 79s because it's in the bromine column, bromine row, and then bromine depth here. So this particular box is the probability of having 3 bromine 79s. If I want to have 2 bromine 79s and 1 bromine 81, that would correspond to, for example, this box here. And also, I notice if I come down bromine 79, bromine 81, this would also correspond to that. And then last but not least, I also have a box in the back of 79, 79, 81, and here. If I go through this and actually determine the identity of all the boxes, I'm going to get an interesting situation. I'm going to get that for 3, the probability of 3 bromine 79s is going to be 1 block out of a total of 8, so the probability is 1/8. The probability of 2 bromine 79s and 1 bromine 81, that will end up corresponding to 3 blocks, so the probability there is going to be 3/8. The probability of 1 bromine 79 and 2 bromine 81s is again going to be 3 out of 8. And then last but not least, the probability of 3 by 81 bromine is going to be 1/8. So we get a relative proportion of 1 to 3 to 3 to 8. And the masses that go along with that, so the peak heights, the peak positions, so this would correspond to the peak heights. We'll get a ratio of 1 to 3 to 3 to 1. And then the masses that go with those, this would correspond to the M, the molecular peak. The second situation would be the M plus 2 peak. The third situation would be the M plus 4 peak. And then last but not least, the combination for 3 bromine 81s would be the M plus 6 peak.
Here we have a 3 by 3 Punnett cube corresponding to the situation of having exactly 3 bromine atoms in a single molecule. So this particular column here would be bromine 79. Here would be bromine 81. Again, in this direction, bromine 79, bromine 81, and then up and down, the top is bromine 79, and the bottom is bromine 81. Relative to the figure in the JChem Ed paper, this is a rotation of the cube so that we can see this back cube a little bit better. But we have the situation where the pure white cube corresponds to 3 bromine 79 atoms. The striped boxes correspond to the probability of having 2 bromine 79s and 1 bromine 81. The situation of having this light gray is the situation of having 1 bromine 79 and 2 bromine 81s. And last but not least, the darker gray is the situation of having 3 bromine 81s. Then to assess the overall probabilities, we are going to be proportional to the volumes, and we have to sort the cubes by the color. So remember, this was the case of 3 bromine 79s. Next we have the case of 2 bromine 79s and 1 bromine 81. Then we have the case of 1 bromine 79 and 2 bromine 81s. And then last but not least, we have the case of 3 bromine 81s. So the relative proportions are going to be 1 to 3 to 3 to 1. Or, since we have a total of 8 cubes in general, the probability of 3 bromine 79s is going to be 1/8. The probability of 3 bromine 81s is going to be 1/8. The probability of 2 79s and 1 81 is going to be 3/8. And the probability of 1 bromine 79 and 2 bromine 81s is going to be 3/8, approximately.
Chlorine exists in 2 isotopes, the same way that bromine does, but instead of having a 1-to-1 relative proportion, the relative ratios are roughly 3-to-1 in the case of chlorine. So we can draw a Punnett square box for chlorine, and we'll make one column for chlorine 35 and a second for chlorine 37. These are the 2 naturally occurring isotopes of chlorine. And as a crude sense, we're going to label them with the relative probabilities. It's a 3-to-1 ratio, so let's just use 3 and 1. That's what we're going to put there. So we want to look at what is the probabilities that we're going to get certain combinations of chlorine isotopes in a molecule that has exactly 2 chlorine atoms. A typical molecule for which this would be true would be methylene chloride. So we can just simply multiply the values for each column. So 3 times 3 gives me 9. 3 times 1 gives me 3. 3 times 1 gives me 3. And 1 times 1 gives me 1. And again, we notice that this particular box here is for 2 chlorine 35s. These two boxes are each 1 chlorine 35 and 137. And then this box down here is the combination for exactly 2 chlorine 37s. So one thing again we want to do just to make sure we calculate our probabilities accurately is to highlight these two boxes, because these two boxes correspond to equivalent circumstances. So we have to add those probabilities together. And what we get is that the relative ratios are going to be, so we have for 2 chlorine 35s, the probability, so we add up all the numbers in here, so it's 9 plus 3 plus 3 plus 1 gives us a total of 16. So the probability of just 2 chlorine 35s is going to be 9 out of 16. The probability of one of each is going to be 3 plus 3, 6, 6 out of 16. And then the probability of 2 chlorine 37s is going to be 1 out of 16. And the important thing to notice is the ratio is going to be 9 to 6 to 1. And this is the classic ratio of peak height that we might see in mass spectrometry, which would suggest to us that our molecule has exactly 2 chlorine atoms. The 9 would correspond to the M molecular peak. The 6 would be the height for the M plus 2 peak. And then the 1 would be the M plus 4 peak. Now what we can do with this table, beyond what we've done so far, is to actually make the width of columns and the height of rows proportional to the probabilities of a particular isotope. So let's look at how we might do that. And we're also going to combine it with the high resolution, the exact numbers that we get from the isotopic distribution. So drawing our, slide this over a little bit. And I'm drawing it here roughly 3 to 1. But for high accuracy work, we can use a painting program to actually measure it out exactly. And we can actually turn it into a graphic if we like. So this is the probability for chlorine 35. This is chlorine 37. And then we can use the exact values for the isotopic probabilities. So this one is 0.7578. And then the probability for chlorine 37 is 0.2422. And then for over here we have chlorine 35 again. And we know the probability is 0.7578. And then for chlorine 37, it's going to be 0.2422. So we've combined several things into this Punnett square. Instead of using just a basic 3 to 1 ratio, we're actually using the exact probabilities. Also notice that if we add up the probabilities along for the columns, it adds up to 1. And then if we add up the probabilities for the rows, it adds up to 1. That will give us a normalized probability distribution when we calculate. Because if we calculate 1 times 1, we get 1 total, which is what we'd expect for the overall probabilities. And now we can actually just fill in the entries into each box based upon multiplying the value of the row with the row of the column. And let's see what the exact values we get when we do that. This ends up being 0.5743. Rounding these a little bit, so if they don't add up exactly to 100, that's why. 0.1835. 0.1835. And then 0.0590. So here we actually have the more exact ratios. So the probability of having 2 chlorine 35s is about 57%. The probability of having 2 chlorine 37s is just a little less than 6%. And we also notice that the areas of each box, if we actually perfectly carefully, is actually proportional to the relative probabilities. So again, if we normalize these, we would realize that their relative ratios are going to be approximately 9 to 6 to 1, which is the situation with which we're familiar with 2 chlorine atoms.
Here we have a 2 by 2 Punnett square corresponding to the situation of having exactly 2 chlorine atoms in one molecule. We can envision the left box, which is this column here, as being the probability of having a chlorine 35. This second column, which is narrower, is the probability of having a chlorine 37. And the approximate ratios of their widths are 3 to 1, which are exactly the ratio of probabilities of the isotopes. Similarly, for row 1, notice that row 1 is 3 times as tall as row 2 is. Row 1 corresponds to the second chlorine atom being a chlorine 35, and this corresponds to the probability in the second row of having a chlorine 37. So therefore, this large box in white is the probability of having exactly 2 chlorine 35s. The light gray correspond to the probability of having 1 chlorine 35 and 1 chlorine 37. And last but not least, the darker gray square corresponds to the probability of having exactly 2 chlorine 37s. And to assess the overall probabilities, we have to add together the areas of like-colored boxes. So if we make a kind of shift here, we can see that this will make it easy to add the areas together. We notice that the relative areas are going to be 9 to 6 to 1. And this is exactly the ratios that we get for peak heights for M, M plus 2, and M plus 4 when we have a molecule in mass spectrometry that has exactly 2 chlorine atoms. Here is a model of a 3 by 3 Punnett cube corresponding to the situation of having exactly 3 chlorine atoms in one molecule, as in, for example, chloroform, CHCl3. In this direction, this column is chlorine 35. This is chlorine 37. Therefore, we have a 3 to 1 ratio in the lengths. In this direction, this column corresponds to chlorine 35. This corresponds to chlorine 37. And then as far as altitude goes, the top 3 to 1 ratio, so 3 times as tall as the bottom, this corresponds to chlorine 35, and this corresponds to chlorine 37. So therefore we end up with a situation where we have this large white box here is the probability of having 3 chlorine 35s. These light gray boxes here, so this one essentially has a 3 by 3 by 3 volume. If we consider the length here to be 3 and the length here to be 1, this has a volume of 3 by 3 by 3. That's the 3 chlorine 35s. This box, and another box which is underneath here, it's a little harder to see. These correspond to the situation of having 2 chlorine 35s and 1 chlorine 37. And their areas work out to be 1 by 3 by 3. Here we have in a darker gray, this is the probability of having 1 chlorine 35 and 2 chlorine 37s. There's another one of these here and then on the back side. So there's 3 of those particular types of boxes. And all the way down in the corner here, it's very difficult to see. We have in black the probability of having 3 chlorine 37s. To assess the relative probabilities, we need to add together the volumes of the various like-colored boxes. So let's do that. So again, here we can see visually that this box is the probability of 3 chlorine 35s. These all together add up to the probability of having 1 chlorine 35 and 2 37s. These 3 boxes together is the total probability of having 1 chlorine 35 and 2 chlorine 37s. And then last but not least, the small box in the corner here is the probability of having 3 chlorine 37s. So we can assess the over probability because we know that the entire cube that they formed ultimately was a 4x4 cube with a volume of 64 units. We know that the volume of this box is 3x3x3, which is 27. So the probability of 3 chlorine 35s is 27 divided by 64. The probability of having 2 chlorine 35s and 1 37 is also 27, because we have 3 of these boxes together. So the probability there is going to be 27 divided by 64. For the probability of having 1 chlorine 35 and 2 chlorine 37s, this is, you can recognize as 1/3 the probability here. So the total volume here is 9. So the probability of that particular combination of isotopes is 9 divided by 64. And last but not least, our poor little box here has a volume of 1. That is the likelihood of having exactly 3 chlorine 37s in our molecule, and that is a probability of 1 divided by 64.